All right, we are looking at biological molecules for this chapter. Now I have biological molecules split into two different sections. I have carbohydrates, and fats and phospholipids are going to be this first one. But I'm making it just one big Jamboard because everything relates to each other, and you'll see the table that I want you to be able to fill out in the end. It's the same concepts throughout all of the different biological molecules. So first we are covering carbohydrates, fats, and phospholipids in this lecture, and DNA, RNA, and proteins in the next lecture. So again, make a copy for your Jamboard so that you can move things around without moving stuff around for other people, and then work through. I'll take pauses, and I will allow you to move things around to see if you have an understanding before I give you the answers. Okay, so our first objective here is to look at sugars or carbohydrates. So it really is to diagram or define the structure of sugar. So I will never ask you to draw this again. I just want you to recognize what is a sugar or a carbohydrate. So first of all, just looking at these, what functional groups do you see? And what do you recognize are some isomers of each other? So pause here and put the labels for the functional groups, and then just put the isomer tabs where you think there are two or more isomers. So hopefully you recognize from what we had done before that within these sugar molecules, what you see is that you have a carbon double bound to an oxygen. And if you remember from our functional groups, those are carbonyl groups. So we have some carbonyl groups. So we have some carbonyl groups, and then we have some that are those OH groups. So those are the hydroxyl groups. So carbonyl groups, hydroxyl groups. And then you do see, oh no, not in this one, it's in the other ones. I was going to say you do see methyl groups, but you actually don't see methyl groups down here. There is no CH3 down at the bottom. So carbonyl groups and hydroxyl groups. And another thing you might want to make notes for your notes is that these are basically all carbon chains that have carbonyl groups or hydroxyl groups. And then hopefully what you saw, if you remember, an isomer is something that has the same molecular formula. So if you look in here, can you identify where the molecular formula is? Right, and that's the thing that tells you what's making up the molecule. So here is the molecular formula for both glyceraldehyde and dihydroxyacetone. Those are both C3H6O3. And so those have the same molecular formula, but if you look down here, you can see that there's a different structure. So here, the carbonyl group is on the end. Here the carbonyl group is in the middle. So they have the same number of carbons, they have the same number of hydrogens, and they have the same number of oxygens. However, they have a different structure. So that indicates that those are isomers. And then that happen to be really nice and just kind of list them out for us. So C5H10O5 can either be ribose with the carbonyl group on the end, or ribulose with the carbonyl group in the middle. So also isomers. And then down here, actually all three of these are isomers, all having the same C6H12O6, and then glucose, galactose, fructose, all having different structural formulas. So isomers being same molecular formula, different structure. So just hopefully you see the connection there of how we are taking the information we learned from the previous chapters, applying it to the molecules that we talk about that are important in biology. And when we talk about carbohydrates, carbohydrates are a macromolecule, which just means it's a bigger molecule that's made up of smaller molecules. So carbohydrates are made up of sugars. So the monomer for a carbohydrate is a sugar molecule. So basically, in order to build something larger, you're creating bonds between different sugar molecules. And so when you do that, you build a couple of different types of carbohydrates, or two different types of carbohydrates, or starch versus cellulose. So we'll look how you can take that monomer that is a sugar molecule, right, and then put those together, link them together, create bonds between them to build a larger molecule, which is called a macromolecule. So I've created just a couple of things that I want you to label based on the images that are here, right? So I kind of mentioned a little bit, but what's the monomer, or what are the monomers for starch and cellulose? And how would you label the polymer? What would be the polymer? And then if you look at the similarities and differences between starch and cellulose, what do you notice about that? And then also, if you happen to know the function of starch and cellulose, those are the green ones here, right? One is for structure and the other one is for storage. So take a pause here and go through and see if you can match those up. So when we are thinking about monomers versus polymers, right? Mono means one. So the monomer that is for cellulose is beta glucose, when in contrast the monomer for starch is alpha glucose. And we'll look at that in just a second. So I do want to say, since we're right here, right, the idea is that this is the same molecular structure, right? So it's the same molecular structure, except when it goes to form the ring, it makes a different structure in a ring structure. So same linear structure. However, when it goes to close the loop and make the ring, something different happens. So it changes the monomer. So we can have two different monomers, alpha glucose versus beta glucose, using the same linear structure. So these are the linear structure for both of these is the same, but our ring structure for both of these is different. So just real quickly, those are the monomers. When you put more than one monomer together, then you have what is called a polymer. So mono means one, poly means many. So you have a polymer when you have just a large stretch of things stuck together. And you'll see that for all of the structures that we talked about in terms of carbohydrates, RNA, DNA, and proteins, those are all larger polymers that are created from monomers. And then, so now if we look at the kind of the structure of these, what you can see is that alpha glucose, this hydroxyl group down here, is on the bottom, on the first carbon here. And again, just remember any junction for in organic chemistry, this is a C right here. So that's a carbon. So on this first carbon, the hydroxyl group is on the bottom. And then for beta glucose, the hydroxyl group is on the top. So that is the biggest difference. Well, the difference that causes the biggest effect between starch and cellulose. So all it is, this becomes a ring structure, and then because of the way the bonds form, then this hydroxyl group is on the top for beta glucose, and it's on the bottom for alpha glucose. Now, when you take those and you create the bonds to form the ring structures, then what happens is that you can see that all of the hydroxyl groups, this yellow highlighted hydroxyl groups here, when you create the bond between the glucose molecules, the alpha glucose molecules, right, those hydroxyl groups all stay on the bottom for starch. So that is another difference. And that difference comes from the structure of alpha glucose. And when you create this bond, then all the hydroxyl groups line up on the bottom. If you look at the cellulose, you'll notice that those hydroxyl groups alternate from being on the bottom versus being on the top. So when you're creating the bond between the monomers, that structure has to go like this for cellulose because you're building with alpha or beta glucose, and then it has to go like this for starch because you're building with alpha glucose. So that structural difference leads to, in the monomer, leads to a structural difference in the polymer. And then cellulose for function, right? Often people recognize that cellulose are in plants, right? And it provides structure in the cell wall. So cellulose is important for structure, and whereas starch, which we know in human biology is important for, or animal biology, is important for storage. So we can break down starch. Humans actually can't break down cellulose, but that's why it's good fiber. So we've recognized the similarities and differences between the structure and the function of starch and cellulose, resulting from the difference in structure in alpha glucose versus beta glucose. And now I'm going to show you why cellulose is able to be a structural support molecule when starch is not, because of this alteration in the hydroxyl groups here. So if we go to the next Jamboard, you'll see the cellulose molecule, right? So we have cellulose molecules. They're really long chains of beta glucose, and they are linked by different types of bonds. So one thing I want to have you process again, so just with a different image of it, process what is the monomer and what's a polymer. And I happen to label these in orange, and then I drew arrows for the orange, right? So this is a thing, and then this longer thing is a thing. Which one's the monomer, which one's the polymer? And then I have also drawn in green to go along with these three bond types, right? We have this bond here, and then we have these bonds here that are drawn in dots. So I want you to pause here and label those different types of bonds. So hopefully you're able to definitely get the monomer and polymer, right? So if something is a polymer, it's made up of a whole bunch of things. So the whole thing of this is the polymer. So that is this larger bracket here. The whole thing is the polymer because it's made up of many. The monomer is each individual piece that makes those up. So the individual beta glucose ring structures here, those are the monomers. And then when we go to talk about the different types of bonds, if you remember the easy cheat to remember if something was a hydrogen bond or a covalent bond is that covalent bonds are always drawn as lines, whereas hydrogen bonds are always drawn as dots. Also, covalent bonds are always within one molecule, and hydrogen bonds are always between two molecules. So these long chains here are individual molecules, but then those molecules can interact together, specifically because of the hydroxyl group alterations. So if you look at this bond right here, this is a line, so that is a covalent bond because it's sharing electrons. And then it is between carbohydrates. It is a specific type of bond, and it is a covalent, but it has a specific name, and that happens to be glycosidic. So the glycosidic is just a specific name for covalent bonds within these carbohydrates. And then the dotted, the ones that are drawn as dots, those are your hydrogen bonds in between the two molecules. And I'm going to zoom in on that section just real quick. So what you can see is that within this particular cellulose molecule, right, because you have the hydroxyl group down here at one point, and then the hydroxyl group down here at the other point, you can actually have these hydroxyl group here, right? This has, between the oxygen and the hydrogen, that has the oxygen is more electronegative, so that oxygen has a partial negative charge, and the hydrogen has a partial positive charge. Same thing for this hydroxyl group right here. So because these are alternating, you can have hydrogen bonds between two strands. Now for starch, you wouldn't be able to have that because starch would have all of the hydroxyls down on one side and it would not alternate between the two. So that is why cellulose is able to be more of a structural molecule, because it's able to have hydrogen bonds in between two strands. So now that we have that background, what I'd like to do is take a step back and look at the big picture that we are going to be looking at for all types of macromolecules. So for carbohydrates, right, I'd like you to take a second to look through here. What's the monomer that we use? What is the type of bond? And then if you happen to know the synthesis reaction and then the breakdown reaction. So synthesis reaction means what reaction occurs when you are creating a bond, and then breakdown reaction is what reaction occurs when you are breaking a bond. So pause and find the pieces and move them within the carbohydrates section. And so what we learned just a little bit ago is that the monomer for carbohydrates is a sugar. Okay, so a sugar monomer. And then the specific type of bond were glycosidic linkages. And then here, it's kind of in the name up here for these two, where the synthesis reaction is dehydration synthesis. So just based on that name, what do you think happens when a bond is created between two glucose molecules, or really any other molecule? If it's dehydration synthesis, what are we losing there? Right? So dehydration means we've lost a water molecule. And I'll show you what that looks like in just a second. So when you think about breaking a bond or creating a bond, you always want to think about water. So the synthesis reaction is you are removing a water to create a bond. The breakdown reaction is that you are using water, you are introducing water to break a bond. And I am projecting back, and I just make sure that I said that correctly. The synthesis reaction, you are losing water, taking water away, dehydrating it to create a bond, and then you are, for the breakdown reaction, or to break a bond, you are using water to break the bond, so to lyse, right? So lyse means to break. So hydrolysis means to use water to break something. And just to give you a little bit more of a visual on that, looking at this next sheet, where if you have a molecule, right, you often have hydroxyl groups on the ends of those molecules. So what can happen is that this H from here and this OH from here are going to come apart, and then now you just have that oxygen hooking the two of them. So that is the dehydration. You've lost that one water molecule. And now for the hydrolysis, or the breaking of the bond, you're taking that one water molecule and you are adding it back in. So now you've got the H back on one and the hydroxyl group on the other. So dehydration, taking out the water molecule, and hydrolysis, putting the water molecule back in and breaking that bond. So I do want to fix this because I forgot to do that. There we go. All right, fixed it. I want you to predict or calculate the molecular formula of a given polymer. So what I'm going to do is give you a second to think about recognizing this, is that when we are bringing two molecules together, we are losing a water. Think about two glucose molecules and creating a bond between those two. I want you to know what the molecular formula is going to be of that new molecule. This one would happen to be a dimer. This one down here is going to be a polymer because there's more than two. So if you remember, just to get you started, is that the molecular formula for glucose is C6H12O6. So that is your molecular formula for 1. So take a pause and then see if you can come up with the molecular formula for 2. So when you're thinking about this, the easiest thing to do is just to take this and multiply it by 2. So basically you're taking this 6 here, multiplying that by 2, the 12 multiplying it by 2, and the O multiplying it by 2. So that gives us C12H24O2. But we haven't done something. And so if you happen to recognize that when you just take your 2 molecules, right, and you multiply out the carbons, hydrogens, oxygens, you have to always remember that for every bond that you create, you take away 1 molecule. Oh, sorry, one water molecule. So what we need to do out of here is take away that 1 water molecule, and then that would give us C12. This carbons always stay the same because you're not losing any carbons there. But we had 24 hydrogens. Now we're taking away 2. That gives us 22 hydrogens. And we had 12 oxygens, but we lost 1 in that 1 water molecule, so we're going to have now 11 oxygens. So with that knowledge, try the calculating the molecular formula for 4 glucose molecules. So remember, it's C6H12O6. Now you need 4 of it. So now think about how many bonds you need to create, which means how many water molecules you're going to take out. So take a pause and give that a try. So I promise you my handwriting is much nicer than this normally. It's hard to do it on with the mouse. However, what you should come up with is 6 times 4 is 24, 12 times 4 is 48, and 6 times 4 is 24. So C24H48O24. However, that's not the end. If you think about how many water molecules you need to take out, I'm just going to put, we've got a glucose molecule here. We need to bond that, so we've got 4 of them. So let's see how many, and sometimes this is just helpful to do so that you remember. A lot of students will say, well, I have 4 glucose molecules, so I need to take out 4 water molecules. However, the water molecule comes out when you create the bond. So if you think about how many bonds you have, then that's where the water molecules are going, right? So there's 1 bond, 2 bonds, and 3 bonds. So you're making 3 bonds between the 4 glucose water molecules. So how many water molecules are you going to lose from that? All right, if you're making 3 bonds, for each bond you're going to lose 1 water molecule. So now you need to get rid of 3 water molecules. And that happens to be H6O3. So we're going to get rid of 6 hydrogens and 3 oxygen. So that leaves us in the end that 4 glucose molecule. The final molecular formula for that is C24H42O21. And so there are more complicated examples that you could have with different polymers, but this is I'm going to stick to this one just because if you understand the concept, that's all that I really care about, is that you understand that every time that a bond is created, you are losing a water molecule. And I might give you on an exam, I might give you one of these problems to do. So be ready for that, but I won't ask you to do like a more complicated protein synthesis one. I would stick it to the carbohydrates just because it gets too messy with the other ones. So now that we've looked at the carbohydrates and how we form a bond, one of the things I want you to pay attention to and notice is as we're going through these other ones, the lipids, the proteins, and the nucleic acids, keep in mind thinking about what type of monomer, what type of bond is between those monomers, And then just as a spoiler alert, the synthesis reactions are all the same and the hydrolysis reactions are all the same. So if we move on to our next objective, which is to compare and contrast those phospholipids, I again want you to look at this and process the images. So what functional groups do you see? How many fatty acid chains are there in the fats and the phospholipids? And what structures are common between the two? So in this one, what you'll see is that this is the actual final fat molecule, this one on the bottom. This one, they are showing you a dehydration synthesis reaction where you're losing that water molecule to add a fatty acid group onto the glycerol. And then, you're doing that 3 times to create that fat molecule. And then over here is kind of the similar outline. Just take this and rotate it on the side to find the similarities and differences between this fat molecule. So really what you're comparing is this and this right here. So what structures are common between the two, how many fatty acid chains are there, and what functional groups do you see? So take a pause and address that. All right, so hopefully one thing that you've noticed is that in all of these there are some carbonyl groups. So up here, right where those ester linkages are, this is a carbonyl group here, there's a carbonyl group here, and a carbonyl group here. Over in the phospholipid, there's also those carbonyl groups, and they are located right here. So you've got carbonyl groups in both of these. There are some hydroxyl groups, but they are only within this one, so they actually are not in the final molecule. But one thing you will see is that the methyl groups, you do have CH3 methyl groups on the end of those fatty acid chains. And so when you are, oh, and I forgot, there is a phosphate group. I almost forgot that one. So there's a phosphate group right here. So this with the phosphate and the bond structure with the oxygens around it, that's a phosphate group that gives this phosphate head of this particular phospholipid a negative charge. And then what you'll see, this is a little bit different, but just a different representation, right? It's like you're in organic chemistry already, and they're doing those little kink structures where every junction is the carbon and there are still hydrogens. So basically this is just a fatty acid chain, and it looks like this. So there's a little bit difference in here, whereas this fatty acid chain has a double bond in it, which is going to give it a kink. So the similarities is that they both have fatty acid chains, and then there is a difference that in one of these fatty acid chains there is a double bond. But then another thing that they have in common are the ester linkages. So for both the fats and phospholipids, they have these ester linkages. If I can rotate it and like just put it right on that ester linkage right there. So the idea is that the glycerol is present in both of these. So I'm going to add in a common. If you ever need another post-it, always you can just duplicate them if you ever need it. So the glycerol is common across the two, and the ester linkages between the glycerol and the fatty acid chains are common across both of them as well. Now, the difference comes in where the phospholipid does have a kink in that, which gives it specific properties, so it changes the shape of it. That double bond right here between the carbons in this one changes the shape of it. It's not a linear or saturated fatty acid. It is an unsaturated because there are more hydrogens that could be added. And then another big difference is that there is no phosphate group over here on the fatty acid, or on the fat. So basically, in order to get a phospholipid, you take one of the fatty acid chains and you replace that with that phosphate group that's up here. So this phosphate group is different, and that actually gives it very different and important properties that we will look at a little bit later. So one thing to remember is that that phosphate group has a negative charge, And so anything that has a charge to it will want to interact with water. So that's why this says hydrophilic. And then anything that is neutral, which carbon and hydrogen chains, right? Carbon and hydrogen have equal electronegativities. So they're pulling on the electrons equally. So they don't have partial positive or partial negative charges, meaning that they are neutral. And neutral things do not like water. So if you think about a oil or a fat, this is their structure, and so they don't really have charges, and so they don't like to interact with water. However, the phospholipid, because it has that phosphate group, it does have a charge, so part of it does want to interact with water, even though the tails do not want to interact with water. So if we take that concept of the phospholipids, because those are really important in biology, and we recognize that the hydrophobic tails don't want to be around water, and what that allows to be created are membranes. So membranes are really important. If we didn't have cell membranes, we wouldn't have cells. And so our next objective here is to look at the membrane and label the different areas based on charge and neutrality, and then hydrophobic and hydrophilic areas. So we will get into more membrane details later in the membrane chapter, but here, because we're talking about phospholipids, I want you to recognize and remember in the later chapters that those membranes are composed of phospholipids, and it's what we call a phospholipid bilayer, and that the pieces that create the larger membrane are actually what give the membrane its really important properties. So just really quickly, pause here and just tell me what area of the membrane is neutral, what area of the membrane is charged, and then where are those hydrophobic and hydrophilic areas. Okay, so like we just mentioned, the phospholipids have that negative charge, so they are negative charge, so technically we need to make another one of these and put it down here. So 2 parts of the membrane are charged, and the 2 parts that interact with water are the ones that are charged. The ones that are neutral and have no charge do not want to interact with water, so they're kind of come together, and they're like, no, I hate water, and the other one's like, oh, you hate water too, oh good, we're protected on either side by our other friends who actually like water. And so anything that is neutral is going to be the hydrophobic region, so those hydrocarbon chains are hydrophobic, and then the hydrophilic parts are the charged parts. This is the part that actually wants to interact with water, and so we technically have 2 of those. And so we will talk about membranes at a later date. I will add on to your Jamboard a Amoeba Sisters membrane overview in case you want to look at that. But for now, this is all I really need you to know. And then we'll get into membranes more in the next lectures. I'm going to continue on with the macromolecules or biological molecules section, and we'll cover DNA, RNA, and proteins. Oops, except wait a minute, I forgot one thing. Go ahead and fill this out for lipids. So take a second, fill it out for lipids, and then I'll come back and show you. All right, so technically these are not, so the fats and phospholipids are not macromolecules, so technically not really monomers, but the thing that makes them up are glycerol and fatty acids. And then the type of bond between those things are called ester linkages. And then the same thing for synthesis, it's dehydration synthesis, and the breakdown is hydrolysis. All right, this time I promise I'm really done.